All right, welcome back everyone. So we're going to pick up here and we're going to begin looking at platelets, and then we're going to get into the process of what's called hemostasis, and we'll discuss this very important physiological process. So first off, before we talk about hemostasis, we're going to talk about platelets because platelets are involved in the hemostasis process. Hemostasis just means blood clotting. So platelets are, we're going to call them cell fragments, and not necessarily cells because they're derived from a cell, from a large cell called a megakaryocyte, right? But they themselves are not cells. They're from a cell, but they're not cells themselves. So they don't have a nucleus, they don't have organelles like a traditional cell, but they're these fragments of larger cells called megakaryocytes that begin to split into all these smaller fragments. These have a major role to play in blood clotting, in the sealing off of a damaged section of blood vessel. You typically have in a cubic millimeter 150 to 400,000 of these. So you may see it just referred to on a CBC or some other type of clinical panel of blood values, like 150 to 400. Right, below 150 is too low. Above 400 is too high. Right, and so you can imagine, right, what's going to happen if you have too many platelets or too few platelets, right? If you have too many, right, what could happen? Maybe you're at risk for forming clots, right? Additional clots that you don't need. If you're too low, maybe you're at risk for bleeding, right? So again, either way, it can be dangerous. So you want to keep it in a nice kind of steady homeostatic range, right? Many of these are stored in the spleen. They have a short lifespan. They're broken down. They're recycled, just kind of like some of our other cells, like our red blood cells that we've mentioned previously, right? So we'll come back to platelets here in just a moment when we talk about the process of hemostasis, right? So what is hemostasis? If you look at the word hemostasis, the term hemo refers to heme, which is referring to blood. And then stasis, right? If you think of the term static or stasis, this means like to halt, to stop, right? Not moving, right? So hemostasis means to stop the movement of blood or stop bleeding, right? Specifically. The process of hemostasis we can divide into a couple of different phases. First one is called vascular spasm. Second one is called platelet plug formation. And then you have one called coagulation. Right, so vascular spasm, platelet plug formation, and coagulation. Now, in reality, some of these are kind of overlapping. So you may have some vascular spasm occurring along with platelet plug formation, but we're going to talk about them in order: vascular spasm, platelet plug formation, coagulation. Right. So oftentimes you'll hear, I just want to make a little note, you may hear coagulation as blood clotting, and that's what it is. But in reality, coagulation is just one part of blood clotting, right? It's a part of this greater, broader hemostasis process, right? So first stage or first phase is called the vascular spasm, right? So vascular spasm, if you look at the word vascular spasm, vascular means vessel, and spasm in this case is meaning to constrict, right? So vascular spasm occurs when the blood vessel constricts, right? The stimulus for this process is damage to a blood vessel, right? So the blood vessels normally are at some diameter, right? And again, we're dealing here with smooth muscle, right? Smooth muscle arranged in kind of the circular arrangement. So normally, our endothelial cells lining here are endothelial cells lining our blood vessel are helping to keep our smooth muscle cells relaxed, preventing constriction. Whenever you damage the endothelial cell, they release a chemical called endothelin, and that endothelin stimulates the adjacent and nearby smooth muscle to begin to constrict, and so you end up with this constriction of a blood vessel, right? So again, it's going to go from one size and it's going to decrease in diameter. And so you can imagine what the purpose of this is, right? Why would, when you damage a blood vessel, you cut your finger, you have some other type of vessel damage, why would you want to constrict the vessel, right? Well, largely, if you constrict the vessel, maybe we're reducing the amount of blood that's flowing to that area, and so you reduce blood loss, right? And so that's kind of the main initial acute, very sudden, very quick response, right? Is what's called vascular spasm, right? Second one is called platelet plug formation, right? Platelet plug formation. So with this, again, the name of the phase tells you what's going on. We have platelets beginning to clump together, right? So by forming this plug, we're preventing, again, additional blood loss. So you can imagine that we need to, during normal times, right? Normal times meaning when we don't have vessel injury, vessel damage, you want to keep the platelets inactive, right? You want to inhibit them, right? You want to inhibit your platelets. So normally, when we don't have tissue damage, our vessels are coated with this chemical called prostacyclin, which is a prostaglandin, and this reduces or prevents platelets from clumping together. Right? It prevents them from sticking together. Right? Which again, under normal cases, normal non-injury context, that's good. Right? That's what we want. So when you damage a blood vessel, then now we want to activate the platelets, right? We want to bring them together and form a clot, right? So one thing that happens is you damage the vessel and you expose collagen fibers in the wall, right? So this inner layer, you've got endothelial cells, here you've got smooth muscle cells, and then all out here on this outside, you've got these collagen fibers, right? Collagen fibers, or the endothelial cells too, will result in the production or the exposure of this molecule called von Willebrand factor, right? This VWF, von Willebrand factor. What von Willebrand factor does is it provides an attachment site for platelets. So platelets that are now floating by will begin to stick as they become activated. Platelets will stick to this von Willebrand factor, right? The von Willebrand factor again binds these platelets, the platelets stick, the platelets then form these long kind of cellular extensions that then allow them to grab onto other von Willebrand factor molecules, but also grab onto each other. So now you're forming kind of these cross links between adjacent platelets, right? And then you begin to have the activation of those platelets. So the activation of the platelets is essentially the platelets release of so they're activated, but then they begin to release these chemicals called ADP, adenosine diphosphate, thromboxane, A2. We're not going to spend too much time on this, but what this is doing is these are essentially molecules that are then going out and recruiting additional platelets to the site of injury, right? And so when you activate one platelet, it sends out these molecules, it attracts additional platelets that come to the site. Those platelets become activated. The platelets release their chemicals, right? These granules break down and release the chemicals. This attracts additional platelets. Additional platelets come, they bind to each other, they bind to the von Willebrand factor, they clump together, and this process continues and continues, right? Now, one important thing is that the activation of these platelets is local, okay? Meaning that you damage a blood vessel in a certain area and you're activating platelets only that are in that given area, right? So why might this be beneficial, all right? Why is this necessary to activate the platelets only in a small area? Well, the reason is because you don't want to throw clots. You don't want to begin forming clots all throughout the body, right? You want it only in the area of the body where you have this tissue injury, right? So again, it's this local process. So you can imagine here this process of recruiting platelets, binding them, activating them, recruiting more, they become active, they recruit more, they become active, and so on. This is a great example of a positive feedback mechanism, right? Again, as opposed to a negative feedback mechanism. So this is one of our few kind of examples or good examples of a negative feedback process, right? We mentioned previously in one of the parts that, or previously in this part about high or low platelet counts, right? So thrombocytopenia, that's too low. You could also have one called thrombocytosis, which is too high, again, affecting clotting in one way or the other. Preventing clotting, causing potential bleeding, or forming too many clots, having too high a platelet count, which could potentially result in the formation of clots. Right. Okay, so vascular spasm, platelet plug, and then the third phase is called coagulation. Right. So coagulation. So this is our blood clotting. Right. Which again is a part of hemostasis. Right. So the overarching idea here of coagulation is you're trying to form something called fibrin. Okay, this fibrin here, that's what we're trying to form. All of this kind of blue turquoise cross meshwork material, all of this is fibrin, right? Fibrin is formed from fibrinogen. When you see that ending ogen here, O-G-E-N, that is meaning it's an inactive form, okay? So you have to convert fibrinogen into fibrin. Fibrinogen is soluble. Fibrin is insoluble, right? Meaning it doesn't dissolve in the blood and the plasma, and it holds the clot, it holds the platelets, it holds the other cells together, right? So you're trapping erythrocytes, leukocytes, platelets, plasma proteins, all in this large clot, which again is helping to minimize blood loss. Right, minimize blood loss. So you have several things that are involved in the coagulation process. Right, many of them are what are called clotting factors. Clotting factors are mostly produced and synthesized in the liver. Many of them, or pretty much all of them, are in an inactive form, so they have to be converted from an inactive form to an active form. Importantly, they are named, or rather they're numbered, according to their order of discovery, and not necessarily the order in which they're involved in this coagulation process, right? Which is unfortunate. If they were named according to their order, it would be easy. Factor 1 would activate factor 2, and factor 2 would activate factor 3, and factor 3 would activate factor 4, and so on, right? Rather, they're named again according to their discovery, so it doesn't flow exactly like that. Okay, there's a joke whenever you look into stuff about coagulation that every year somebody gets to come up with a new part of the coagulation process, right? We get to make something up, right? That's not true, but it's just a funny joke because it's very involved. It's a very complicated process, right? Many of these factors, 2, 7, 9, and 10, require vitamin K for synthesis. So vitamin K is largely, or at least somewhat, made in our gut by bacteria, and so you have to have this bacteria in your gut to make vitamin K. And so who could you think of that may not have bacteria in their gut yet? This can be newborns, right? They haven't been colonized. Their gut hasn't been colonized yet. And so newborns, before they're given injections, they may have to be given another injection with vitamin K. Okay, so to help kind of kickstart the process of making these clotting proteins, and then if the vitamin K is not given, then a period of time has to be you have to wait a period of time to allow the gut to become colonized, bacteria to begin producing vitamin K, and then these vaccines can be given to the newborn. But again, you have to have these clotting factors first, so as to prevent bleeding, things like that, right? So you can imagine you don't want to bleed out from a simple paper cut. You don't want to bleed out from a nosebleed. So forming these clots is incredibly important, right? Now some, again, like mentioned down here, factor 7, factor 9, these are proteases, so they're proteins, and they're going to cause one type of clotting factor, or one form of a clotting factor to be transformed or converted to its active form, right? So we'll see that in a graph here in just a minute, right? That you'll see factor 12 goes from factor 12 to factor 12A, which the A just means activated, right? That's all that means. So there are two pathways, or there's three, but there's two that converge onto one, right? So there's what's called the intrinsic and the extrinsic pathways. and both of these converge onto the common pathway, right? High-level overview, both of these pathways, intrinsic and extrinsic, end with the formation of factor 10, and the common pathway begins with factor 10. Right? So both intrinsic and extrinsic are trying to get to the same point, and then once they get there, you start what's called the common pathway. Right? So I'm going to go over parts of these here really quick, and then I'm going to show you a picture that will hopefully make a little bit more sense. So intrinsic pathway, right? Intrinsic pathway, damage to inside of the blood vessel wall, right? So the interior of a blood vessel for whatever reason, some type of internal trauma, right? This occurs in the first couple of minutes, or it takes a couple of minutes for this process to occur. Easy way to remember this in terms of the numbers of the clotting factors, and the numbers are Roman numerals, right? So the ones involved are 12, 11, skip 10, 9, and 8, right? 12, 11, 9, and 8. So 12 activates 11. 11 activates 9, so you're skipping 10. 9 activates 8, right? So factor 12 converts inactive 11 to active 11. Factor 11 activates inactive 9 to active 9. Factor 9 combines with calcium. Calcium is an important mediator or important molecule involved in the clotting process, so you have to have calcium, along with factor 3 to form a complex that activates inactive 8 to 8. So again, 12, 11, skip 10, 9, and 8, right? You can remember you skip 10 because that's our goal, if you will, right? Our quote-unquote goal. We're trying to get to factor 10. And then that factor 8 activates inactive factor 10 to active factor 10, right? So 12 converts 11, 11 converts 9, 9 converts 8, and 8 converts 10, right? This is intrinsic pathway. Extrinsic is a little bit more simple. This is occurring very quickly, okay, outside of a blood vessel. External trauma, external damage to a blood vessel, right? With this one, easy. Again, you're trying to get to 10. The two factors involved, clotting factor 7, clotting factor 3. So the way you remember this, 7 and 3, you add them together, you get 10, right? This is extrinsic, right? So tissue thromboplastin or tissue factor, this is factor 3. So this here, tissue thromboplastin, that's factor 3, right? This combines with factor 7 along with calcium, that activates factor 10. So factor 3 plus factor 7 and calcium form a complex, and that complex activates factor 10, right? From factor 10, you can then start your common pathway, right? So common pathway starts with factor 10, and it proceeds ultimately until you have formed something called fibrin, right? Until you have formed something called fibrin. So I'm going to go past this slide, and I'm going to show you on the next slide the whole kind of summary of all of the different pathways, right? And then we'll go into the common pathway. So here's our intrinsic that we did first. So again, 11 to 11A. So notice the A, that just means active. 11A converts, 12 converts 11, right? So 12A, 11A. Skip 10, you have 9, right? There's also factor 8 in here, and then from factor 8, you get factor 10, right? So again, you're getting to you're getting to 10A, activated factor 10. Here you see the extrinsic, again, external damage to a blood vessel. Here you have factor 7, and then you have tissue factor, that's factor 3, right? So tissue factor and 3, tissue factor 3, factor 7, again, along with calcium, forms this complex, and this complex converts inactive 10 to activated factor 10, right? Now that you're in the common pathway, it's just one pathway down, 4 main steps, right? So factor 10 converts what's called prothrombin to thrombin, right? And again, these are additional proteins. Thrombin activates fibrinogen to fibrin. Okay, remember our fibrin, the insoluble form. Fibrinogen is the soluble form. And then fibrin, along with factor 12, is helping to stabilize our clot. And again, it's forming kind of that meshwork that stabilizes the platelet plug that we formed in one of our previous parts, right? So here you can see another schematic, another graph showing largely the same thing, right? So again, Intrinsic, so 12, 11, 9, and 8. Extrinsic, 3 and 7, both converting or activating factor 10. Factor 10 activates prothrombin activator, which converts prothrombin to thrombin. Thrombin converts fibrinogen to fibrin. Fibrin, along with factor 12 and calcium, are activating our fibrin, right? Or fibrin, along with it, is helping to stabilize our clot with the fibrin, right? So that kind of meshwork that lies over a clot to help stabilize it, right? So again, intrinsic pathway, extrinsic pathway. Again, pay attention to which factors are involved. So again, intrinsic 12, 11, 9, and 8. Extrinsic 3 and 7. Again, you have calcium involved with both. Remember, you need vitamin K for certain factors. And then they both end with common pathway with factor 10, and then the common pathway begins with factor 10 and goes all the way till you create fibrin. Okay, so let's stop here, and then we'll pick up with our last part, talking about a few additional things related to coagulation.